Today I will be discussing synaptic communication. Synaptic communication is used for crisis management. Synapses can form between two neurons, a neuron in a muscle cell, or a neuron in a gland cell. In this video, I will show synaptic communication between two neurons. The axon, which is up here, of a transmitting neuron has an action potential, represented by the purple arrow, moving down the axon terminal of a presynaptic neuron. The presynaptic neuron is transmitting signals to the postsynaptic neuron at the bottom. This is the presynaptic membrane, and this is the postsynaptic membrane, which is represented by red licorice. The action potential moves down to the L-type voltage-gated calcium ion channel, represented by the green Smarty. The channel will open when the membrane potential changes. The calcium has lower concentration inside the axon terminal compared to outside. So when the channel opens, simple diffusion happens and the calcium ions, which are the broken yellow Smarties, go into the neuron. The calcium ions cause the synaptic vesicles, which are the brown rings, and causes exocytosis. When the neurotransmitters, which are the purple nerds, leave the vesicles, they go into the synaptic cleft, which is called propagation. The synaptic cleft is the space between the two neurons, and it is filled with extracellular fluid, which is blue, and the white is intracellular fluid. The neurotransmitters will then go to the ionotropic receptors, which is the top of the Twix, and go through the ligand-gated ion channels, which is the rest of the Twix. This causes ions to enter or leave the neuron. If there is enough sodium ions, which are the pink nerds, entering the neuron, then depolarization will occur. If the graded potential is great enough in the postsynaptic membrane, then it might be enough to reach threshold and create a new action potential, but there's also chances this might not happen. If the graded potential slightly depolarizes the membrane and doesn't reach threshold, then it causes EPSP, which is excitatory postsynaptic potential. However, when the postsynaptic membrane potential becomes more negative, which is shown, ion channels open and inhibits another action potential forming. IPSP happens, which is inhibitory postsynaptic potential.